We've completed the components of a virus, the definition of a virion, as well as the viral life cycle. So now we're going to talk a little bit about how we would actually grow viruses in a research-like setting. Remember that they are intracellular obligate parasites, which means they can't just simply be grown on an agar plate like we can do with bacteria. So the two main ways, or the simplest way, especially with my experience in viral research, is to do culturing, also known as tissue culturing. And a couple others we'll talk about in a minute, but basically, by definition, when you tissue culture something, you take a bunch of cells or tissue, it's in some type of artificial medium, which is like a broth. It is found in a flask of this sort. The tissue is minced up into small little pieces, and then the virus is grown in that. On a regular basis, the liquid media has to be changed because just like when you're talking about bacteria on a plate, it provides oxygen and nutrients, and that tissue needs to remain alive. The goal while you're growing this, and you would check this under a microscope, remember you can't use a light microscope, so it would be either through fluorescence or an electron microscope, is you're looking for one confluent monolayer. If you allow a tube of tissue culture with viruses to overgrow, the waste products overtake the nutrients found in the broth for the tissue culture to actually work. So this culture is inoculated on a regular basis. The media is changed out, and this is done, there's a color indicator similar to phenylalanine where it's pink and then turns yellow like you remember on an MSA plate. If you allow the broth to turn to yellow, the pH has changed and you'll kill off the virus that you are attempting to grow. So when you look at viruses, remember one of the end products that we had talked about before was CPE, which are effects of a viral infection on the host cell. CPE is short for cytopathic effects. So cyto is the cells and pathic are the negative effects on the cells from the viruses. There's two that I would like you to focus on. The first one is inclusions, and the second one is known as syncytia. So inclusions, if you are at a dental visit and the dental hygienist or dental assistant says you have an inclusion on tooth, 11, inclusions are essentially holes. So what you're looking at in this picture right here is there are cells in all these white areas are actually inclusions or holes. This is a virus infecting this particular host cell tissue. So inclusions are essentially holes that are poked by the virus into the cell tissue. This is one of the two cytopathic effects that I need you to know for viruses. And the second one is syncytia. For this, you can think of the word synergy, where the parts are greater, or the whole is greater than the individual parts. If this was a business class, we would be talking about 1 + 1 = 3 of how the teamwork collaboration causes a larger end product. Well, syncytia is when a bunch of viruses group and bunch themselves together and cause these syncytial formations. This is almost a simple way to explain it as a viral version of a biofilm. There is an illness known as RSV, which is respiratory syncytial virus. Respiratory implies that it's a respiratory illness. It is viral in nature, and what happens is in the lung tissue, these syncytia form that causes these large plaques of viruses, and again, that is the second cytopathic effect that viruses can cause. So a little bit more about culturing. Making the flu vaccine that you hear about every single year, these are grown up in an area called the allantoic cavity of eggs, because again, viruses need to be grown in something alive. So flu viruses are cultivated inside of eggs. Oops, sorry about that. And then through a lab process, obviously you do not eat an egg for a flu virus. They turn into shots, and remember that there are many different strains and variations. It's statistical analysis that's actually used to try to determine what strain of the flu is actually going to be hitting that particular year, which is why every year you hear on the news of how effective or ineffective the flu shot happens to be. My grandmother is allergic to albumin, which is egg whites, so she was never able to get the flu shot. So anyone with an allergy to albumin or egg whites isn't able to receive the flu vaccine. So another way that we can actually test and look for viruses is using something known as fluorescent antibodies. So if you were to go to the cdc.org website and look up fluorescent antibody test, you can read about it a little bit. Rabies virus is the example that they use. So what happens is the antibodies are actually fluorescently labeled, and then it's incubated into brain tissue when we're talking about rabies virus, because that virus affects the nervous tissue. And then the antigen and antibody have an interaction. You do a bunch of washes where you wash away things that don't bind together or are known as unbound. And then using fluorescent microscopy is allowing for a visualization. If you see all of these green spots and these orange spots, this is using fluorescent microscopy. So the entire blackened screen is the tissue culture that you saw when we first began talking about tissue culturing. But then the areas where the antigen and antibody reacted together, that is a positive fluorescent test. So if you were looking for an interaction, in all three of these pictures, you can say there is a positive interaction with antigen and antibodies for this particular viral fluorescent antibody test, because you can see it fluorescing. Another way you can look at them is using an electron microscope, because please remember, you cannot use a compound light microscope that we use for bacteria, because that only measures to the micrometers level, and viruses are measured in nanometers. So influenza is an RNA virus, which you can see as it is glowing for you on this electron microscopy or microscope picture. And another way is using agglutination assays or tests. So when something agglutinates, you're basically saying that it sticks together. So you have red blood cells, and if there's a virus, it's hemagglutination. So the virus is actually sticking to the red blood cells. This is done in one of these, it's known as a 96 well plates. We were supposed to do an ELISA test, which is enzyme-linked immunosorbent assay. This is how AIDS testing is done. And we unfortunately are not going to be able to, but that's probably going to be one of the labs that you take a look at through a video towards the end of class. If it sticks, these wells are then color-coded when you have the antigen-antibody interaction. And if it remains a clear color on the well, that's a negative test. For the ELISA we would have done in class testing for HIV positivity, it would have turned a light blue in color. The color is added as an easy way or means to actually read the results of one of these tests. And please remember, when you're talking about higher level illnesses such as AIDS and HIV, you actually would do this test in duplicate and triplicate for all scientific type of tests. And now we are going to completely switch gears, completely. We're going to talk about prions and viroids. So I'm going to share with you all the information on this one single slide that you're looking at here for prions is equivalent to 8 questions on the exam. So it definitely makes sense for you to remember this. I love prions. They're very interesting and exciting. They are nothing more than itty-bitty pieces of protein. So there's no ribosome, there's no nucleus, there's no endoplasmic reticulum. There's actually absolutely nothing but a small little piece of what's known as a rogue protein. So it's a little protein that is going around causing a lot of problems and wreaking havoc in the body. They cause something known as spongiform encephalopathies. So imagine a cork bearer poking holes into the brain. It literally causes holes in nervous tissue or the brain. These diseases are all fatal, and you need to know the pathogenesis. The pathogenesis is, how does a prion actually cause infection? And the answer is, it's an abnormal form of a protein. So it's based on how it folds. It folds improperly at chromosome 20, and that is the pathogenesis of a prion. And again, a prion is nothing more than a little piece of protein. A couple examples for you are Creutzfeldt-Jakob or mad cow disease. Mad cow disease is also gotten from eating contaminated meat, so you may have heard that in the news. This would have been probably an organism that some of you would have been interested in for your final projects had we been able to continue with those. So if you take a look at this visual, some of the signs and symptoms when someone has spongiform encephalopathy or prion infection is there's brain shrinkage, and the deterioration is incredibly rapid, because remember that they are all fatal. And this picture is to show you that, again, the pathogenesis is nothing more than abnormal folding at chromosome 20. This is all written out for you on the previous slide. If you take a look at this section or histology slide of a patient that has Creutzfeldt-Jakob, all of these white areas are holes that are forming inside of the brain nervous tissue of this patient. The picture on the right, you don't have to read all of these things on the side. I just want for you to all see it. The picture on the right is a neuron because a prion infection impacts the nervous system, and neurons are the cells of the nervous system. So NP stands for normal protein. All of these little green cone-looking things are normal protein, and then enters one prion protein, which is PP for short. Notice how it's going to attach itself to a normal protein, and in no time at all, it starts converting all of the normal ones into abnormal ones, and finally, every single protein in the brain has been taken over by a prion, which is why all of these infections are fatal. Next, we have viroids. I have no big interest in these because they relate to plants, which you guys should know by now is not really my thing. I would like for you to not mix this up with a virion. Ooh, that's really sloppy. Hold on a second. With a virion, because please remember from the first part of the PowerPoint, a virion is a completely assembled virus particle. And that is not what we are talking about right now. We are actually talking about viroids. So just like a prion is a piece of protein, a viroid is nothing more than a single circular piece of RNA. There is no nucleus, there is no nucleoid, again, no Golgi, no endoplasmic reticulum. It is nothing but a tiny single circular piece of RNA. It does not, this is very important you remember, it does not code for protein. So it doesn't do much of anything. It's this single circular small piece of RNA that is known to cause plant diseases. And again, you need to know the pathogenesis for this guy, and it interferes with translation because, again, it does not code for a protein. So if you take a look at this picture, this guy down here is known as tobacco mosaic disease. This is a disease in plants, and notice how it's much smaller and it is killing off the plant. And because it's a plant, I'm not really going to speak of it, but I do want you to look at this. This is a smallpox virus and polio, and look at how tiny a viroid is, because again, it's nothing but a small circular tiny piece of RNA, and the pathogenesis is that it interferes with translation, which makes protein, so it does not complete the process. So virus and host interactions, these three terms you should all be pretty familiar with. Acute viruses, when an acute virus is occurring, very, very quickly, the symptoms are going to develop and you hit a climax quickly. Climax is the top peak where all of the symptoms come out when somebody has a viral infection. The example for acute is hepatitis A. Chronic type of virus host interactions are different. They develop very slowly and then they stick around or linger for a long time. There is a long convalescence, which is it's a long period of recuperating, and something known as viral shedding is occurring. So to explain viral shedding, that means that the virus is in fact replicating. It's found in the body in very low numbers. So therefore, during the time of viral shedding, when it isn't in the middle of what we refer to in virus infections as an outbreak, a person may in fact test temporarily negative, but the viral shedding is occurring. And yes, that person can in fact be infectious during that time. And the last term is latent. And I'm sorry, I forgot to mention for chronic, hepatitis B is one of the examples that will help you remember when we talk about that later. Now, the last one is latent. The example for latent is HSV, which is herpes simplex virus, which we'll talk about in the next set of PowerPoints. This is where viral shedding could be occurring, but it's actually not detectable. So here, there's no extracellular virions occurring, but there is viral shedding. Another way to look at latent is that it is sleeping. So somebody with herpes virus could not have an outbreak or visual rash patterns and little carbuncle type of pussy little openings and that obvious rash that goes with the STI of herpes simplex virus, but that doesn't mean that they don't have it. It's sleeping or dormant or latent at the time. So next we need to talk or look at these particular terms in a visual way. Please don't worry about this picture on the right. Just focus on the two graphs because I need for you to understand what you're seeing on them. So remember, an acute infection is one where the onslaught of symptoms and the climax of the symptoms happens very quickly. So the x-axis is time. It's in days because we're talking about measles, which has a quick incubation period. And the other axis is the symptoms that are appearing when someone has the infectious virus. So notice that it's infectious at this point. Here's the little chart for you. You have the disease during this time, and then just as quickly as it was there, it starts to go down. So there's a term that I need you all to be familiar with. It's pronounced sequelae. It's written right here. So when something is sequelae, this is a condition that is a consequence of a disease or an injury that someone has previously had. So there are secondary infections, and here is an example for you. In the bottom picture, again, measles is acute, so quick onslaught of symptoms, the patient is diseased, and then the symptoms go away. So at this point, the patient is non-infectious. But then sequelae shows up, and their SSPE, it's written out for you right here, is a secondary type of sequelae infection related to measles. So the next one, which is chronic, also known as persistent versus latent. Again, please don't worry about this. Just take a look at the charts. So we have the first one of chronic. Hepatitis B is an example of that. So you have the infection, the person is diseased, and notice for a lengthy amount of time, the person is still infectious. This is a visual example of a chronic infection. When something is latent, you have onslaught of symptoms, you have the disease, and then the symptoms go down. At this point, sequelae can show up again, and there's some type of virus activation, and then that person's secondary can go on to have, let's say, shingles. Can you have chickenpox more than once? Yes. Can you have shingles more than once? Absolutely. When you hear something like virus activation, anything that impacts your immune system negatively or causes your immune system to be working when cortisol levels are up, for example, during finals, when you're a little bit more stressed, you have a higher likelihood of getting some type of infection, disease, secondary infection that could be considered a virus activator. Last, we have a very slow infection because, again, you always need to look at incubation times, whether you're talking about viruses or bacteria. So HIV infection, you have the onslaught of symptoms, the disease, and then, again, the lowering of the symptoms. Notice at this point, the axis timeline is in days. But then in years, there's a very slow climb until a person potentially then transitions into having AIDS, which is acquired immune deficiency syndrome. Someone with AIDS will actually die from something even as simple as the common cold, because what's occurring is the B and T cells are almost non-functional, and they're at such low numbers that the body cannot fight off any type of infection. So I just want to mention again one more time that sequelae is essentially a condition that's a consequence of some disease or injury that someone has previously had. So if you want to talk about it in terms of disease, shingles can be sequelae to chickenpox. And if you want to talk about it in terms of injury, a person having poor blood flow as a result of diabetes is also an example of sequelae as it relates to a disease.